Good morning to everyone. Today we will discuss about the central dogma of life, which comes under our biochemistry subject. So what is particularly this central dogma? Firstly, we should know what is central dogma of the life. If you remember, then already I have discussed a little bit about the central dogma of life in one slide. What is central dogma? Central dogma, wait, this particular central dogma, this one is the process, as you can see, this process. Okay, this process. So this central dogma is the process by which the genetic information flows from DNA to RNA and RNA to make a functional product protein. Okay, again I'm saying this one is the process in which the genetic information flows from the DNA to RNA and RNA to finally protein. So this one is called as central dogma, and this process just explains about the instruction in the DNA which are converted into our final product. That one is our protein, and this particular thing is proposed in 1958 by Francis Crick, and I think most of you have listened about this particular scientist name. That scientist name is Francis and Crick, okay, which discovered the structure of DNA. Just remember I had told you regarding the Watson and Crick model, right? So there you the Watson and Crick model, similarly we can say that this particularly central dogma of life is also proposed by the scientist name, that one is Francis Crick. Okay, now this particularly central dogma, it illustrates that the flow of genetic information in the cell, the DNA replication, firstly this particularly DNA replication, as I told you in the yesterday class, DNA replication, and then the coding for RNA, then the coding for RNA, which type of coding? Genetic codon. Okay, the coding for RNA through the transcription process, and further this RNA codes for the protein by translation process. Okay, so the concept of the sequence of interaction can be understood through this particularly framework only. Okay, the most common include the biopolymers, like this particularly our protein. Okay, so this thing you have to remember always, that what is central dogma of the life. Okay, and who proposed this particularly central dogma. Okay, now hopefully if someone asks you what is central dogma of the life, then you have to say what is central dogma, this thing. Okay, this one is the process, this was the flow process is called as central dogma of the life. Already we have discussed regarding the our replication or duplication. Today we will discuss about the revision of transcription and the translation. Okay, so in the yesterday classes, some of you have having a confusion that only I have to learn only two enzyme name in the DNA replication, like that. So don't forget, in the DNA replication I had told you to learn about a four enzyme name. If you have learned those four enzyme name, then definitely you can write down, and if you learn that four enzyme name, that what are those four enzymes and what are the roles of that four enzymes, then only you will be able to write about the DNA replication. Okay, now that what were the those four particularly enzyme in the DNA replication, I had told you helicase enzyme for the cutting purpose, right? The next one after helicase, next one is a polymerase enzyme, primase enzyme, which used to produce the primer only. The next one DNA polymerase, which is used to make a new DNA strand. The last one a ligase enzyme, which is used to just for the ligand purpose, for the ligand purpose, something for the addition. Okay, now come to the point about the transcription. What is transcription procedure? So already I have told you transcription means from DNA to RNA formation is called as DNA transcription process. Okay, and in this as in the DNA replication, that particularly thing is going on through the enzyme named spatial enzyme was a DNA polymerase enzyme. So here similarly in this case, that one is transcription, you have to learn RNA polymerase enzyme. There this important enzyme was DNA polymerase, and here for the synthesis of RNA you have to learn the enzyme name, that one is RNA polymerase enzyme. Okay, now come to the point. What is transcription and translation? Today we will finish or just some revise both things. Transcription and the translation. Transcription means DNA to RNA. Translation means RNA to protein. As I told you, this one double helix model DNA. RNA means single helix. And protein means with this from this particularly RNA, this our protein is synthesized. So now what is RNA? I will tell you a small basic thing. If you have written all those basic things, then this unit will be so easy for you. So firstly come to the our transcription. As similarly to replication procedure, this particular transcription and translation having also three-step: initiation, elongation, and termination step. Okay, is it clear? Everything is there. Now, in the initiation step, you have to learn a small thing. The first thing is promoter region. Okay, you have to learn promoter gene. This one is promoter gene. Then you have to learn after promoter RNA polymerase. Okay, and third enzyme name is nothing. Only you have to learn two enzyme name for particularly this transcription process. That one is promoter region and the particularly your RNA polymerase enzyme. Okay, so what happened in the DNA? Sorry, in this particularly RNA synthesis, how happens? So in the initiation process, what does happen? The DNA molecule, this one is our DNA molecule. This DNA molecule is unwind and separate to form a, as you can see, this DNA molecule is unwind like this. This is separate. Hmm? It is separated to form a small open complexes. After this, what happened into, and this one is held due to this particularly promoter region only. Okay, in the initiation system, what happened? There is a one region of promoter region. Through this promoter region, what will be happen? It will be just unwind. After unwinding, what will be happen in the elongation procedure? RNA polymerase, just like this brown, you can say this one is our RNA polymerase enzyme. This RNA polymerase moves along to the this particular template strand and synthesizing our new particularly messenger RNA. If the particular strand, DNA strand having a dimension 3 dash to 5 dash, so this particularly RNA will make our new this particular RNA polymerase enzyme, it will make our new standard, that one is 5 dash to 3 dash. Okay, and this RNA polymerase will continue make this particularly new RNA until it reaches the termination codon or stop codon. Okay, is it clear? So only you have to learn in the initiation process, only there will be one promoter region. From that promoter region only RNA polymerase come. That RNA polymerase will unbind this particularly DNA. After unwinding, what will be happen? It will make a new RNA molecule, and this process will be continue until it reaches the termination codon. As you can see, until it reaches the termination codon. Now we will see in the particular video. So now we will be able to understand what is particularly thing. Now just see, this one is our RNA polymerase enzyme. As you can see, this one is our RNA polymerase enzyme, right? And what is this? This one is our DNA molecule. Okay, this one is RNA polymerase, this one is our DNA molecule. Now what will be happen? New polynucleotide, because of this RNA polymerase enzyme here, unwinding happens. As you can see here, unwinding happens, unwinding of DNA. And now after unwinding, what will be happen? New, what is this red one? Red one indicate that this one is a new polynucleotide chain comes here. Okay, this new polynucleotide chain will come here and it will make a new messenger RNA molecule, as you can see here. Now this one is rewinding of DNA. Okay, you can see this one is ribonucleotide triphosphate. So hopefully is it clear? And this process will be continue until this particularly RNA polymerase will read a termination codon or stop codon. Okay, when it will read a termination codon or stop codon, then suddenly this particularly process will be stopped. Okay, again listen, this one is our DNA molecule. When this particular DNA molecule having a reason one promoter region will bind to, or one promoter gene will bind to this particularly our DNA. Then what will be happen? RNA polymerase will come to this side. RNA polymerase will unwind this particularly DNA. Unwinding, okay, it will unwind this particularly DNA. After doing unwinding, what will be happen? New ribonucleotide will come together and it will make a new RNA molecule. Okay, they are making a new messenger RNA molecule. After making this messenger RNA molecule, yeah, one after this process will be continued till until this particularly RNA polymerase will read a stop codon or termination codon. When it will read a stop codon or termination codon, then suddenly our this particularly transcription process will be stopped. Okay, so hopefully now is it clear for you? Okay, like this. Now you can see this particular messenger RNA, it will be just eliminate like this. It will be just eliminate this RNA polymerase. This one is just going to this side and this particularly RNA. Yeah. Okay, so this one was our about a transcription procedure. Okay, now come to our next thing. Next thing is our translation process. Hopefully understood within some minutes that what is transcription. Only promoter RNA polymerase. This RNA polymerase will firstly unwind the DNA. After unwinding, new polynucleotide chain will come. It will make a new messenger RNA molecule. After making new messenger RNA molecule, what will be happen? This process will be continued, which one is called as elongation procedure. And after doing this thing, this RNA polymerase, when it will read our stop codon, then suddenly this process will be stopped. Okay, is it clear? This one was so easy. Now come the next one is translation procedure. What is translation? So again, this one is written about our central dogma of the life. So translation means when this particularly RNA to protein synthesis. Okay, RNA to protein synthesis is called as RNA to protein synthesis is called as our translation procedure. Okay, so already we have discussed regarding the DNA formation. Okay, okay, DNA to DNA formation is called as replication. DNA to RNA is called as transcription, and now RNA to protein is called as translation procedure. Okay, now in this particular translation procedure, there is a need to learn mainly three things. Three things, if we have learned, then surely everything will be so easy for you. In these three things, yeah, these three things, first one is messenger RNA, tRNA, and ribosome. Again, I am repeating it. This ribosome having a this. What is this? This one is three site is there. One will be A site. Sorry, this one, the right hand side here, this particular site. Let me take one highlighter. Then yeah, this particular slide is our A site. What is okay? This one is will not be work. No problem. This particular site will be called as A site. Okay, A site, P site, and this one is E site. Okay, E means elimination, P means peptide formation, and this particularly A site means our acceptor region. The acceptor where the tRNA will come will bring a one amino acid. That will bring a one amino acid. That one is called as our tRNA molecule. That one region is called as acceptor region in the peptide form, and now this particularly tRNA, it will just shift to this particularly P site. Now what will be happen? A new tRNA will come here with a new amino acid. The both amino acid will join with each other, and this particularly tRNA will shift to the E side through which it will be just exit. Okay. Now with this messenger RNA is single stranded, which having already we have prepared this one. Okay. Now the next thing, ribosome having us 160S and 40S. What is this SSS? S means this one, Swedberg unit. S indicates a Swedberg unit. One will be called as larger subunit and this one will be called as small subunit of ribosome. E side, P side and A means acceptor, P means peptide, E means exit. Okay, come next. What is this? They are indicating that this particular DNA having a inside, they having a base plus sugar molecule. This one base and sugar. This phosphate molecule, it will be had just outside of this helix, which I had told you in the yesterday class where I have discussed regarding the DNA replication. There only I have discussed that base and sugar will be found at the inside of this particular DNA helix, and base and sugar and the phosphate will be found at the outside of particular helix. Base and sugar is called as nucleoside and base, sugar and phosphate is called as nucleotide. Now in the RNA having a different type of codon. What is codon? I have told you codons is a triplet form of codes which indicate a different amino acid. Okay, and the tRNA will having a tRNA having also code, but that code is called as anticodon. Okay, that codon is called as anticodon. So those codes which are present in tRNA is called as anticodon. Those codes which are present in the messenger RNA is called as genetic code. Okay. Now what is codon? They have written the sequence of the specific amino acid like this. They are having in a triplet form. So one triplet form will indicate a different amino acid. Now you can see this one will be our messenger RNA having a codon, and this particular tRNA having a codes, that codes are called as anticodon. Okay, this one initiation, in the elongation, the termination. This one ribosomal unit having a tRNA, messenger RNA molecule, tRNA having amino acid. Okay, now again this particular translation is also proceeded through a three-step process. First one is our initiation, then elongation procedure, and the next one is termination procedure. Okay, initiation, elongation, and termination process. So initiation means uh, whenever any reaction is just started, that one is called as initiation. And the elongation means whenever the reaction will be proceed, and the termination means whenever the reaction will be stopped. So for this particularly translation procedure, as I had told you, they're having a two types of code. Those codes which are present in our messenger RNA, that codes are called as genetic codes. Sorry, that codes are called as genetic code or codons. And the codes which are present in the tRNA, this greener will represent a tRNA. So that particular tRNA having a codes, that codes are called as anticodon. This thing should be clear. The next thing, you should learn two things. You should have a learn two things. First one is what is this particularly start codon. Okay, this particularly start codon name you should learn. That start codon means AUG and GUG. Whenever it will read up two codons AUG and GUG, then only our reaction will be start. So AUG, GUG, or in my opinion, AUG means AOG and JAOG, GUG. So easily we can learn. Now think the termination codon. Having a termination codons are different in compared to initiation codon as well as this particularly AUG is also indicator our one amino acid too. The next thing, the termination codon having a termination codon, you can see that termination codons are three types. That one is UAA, UAG, and UGA. According to me, for learning purpose, I have done like this. Ua, ug, uga. Okay, ua, ug, uga. UAA, UAG, and UGA. So I think we can learn easily about this particularly codons. Otherwise, if you didn't learn, then someone will write a AUG, someone will write a CUA, and like that. So it will be better if we learn like this only. So what will be happen in the initiation? So in the initiation, first I'm going to tell you. After that, I will show one video. In the initiation codon, the will in this initiation step, what will be happen? This one is our one messenger RNA. Messenger RNA having a codons. Now what will be happen? This messenger RNA, it will be held on this particularly, on this particular messenger RNA. It will be found on this particularly smaller subunit of particularly ribosome. Now what will be happen? Now the another, it means now what will happen? The another tRNA will come with a new amino acid. That one is a methionine having a anticodon. When this tRNA will match its anticodon to this particular codon, as you know very well, A opposite U opposite A, C opposite cytosine, guanine. These are opposite to each other. That means we can say these are anticodons. Now these particular codons and anticodons are just cover up with a larger subunit of a ribosome. It means the smaller subunit of a ribosome having a messenger RNA, and the above that one, it will be covered with a larger subunit of particularly this ribosomal unit. Now when the tRNA will come, it will match our particularly initiation codon, because the initiation will start only through a initiation codon. Now what will be happen? Now this process will be continue. How it will be continue? So firstly, as I told you, this particularly larger subunit having a 3-type site, A site, P site, and E site. So already one particularly tRNA have come at this particularly A site methionine, right? Now it will be, because this one is the starting particularly tRNA with another amino acid. What will happen? This tRNA will shift to the P site. Now another tRNA will come here and it will have a different amino acid. Now what will be happen at the P site? What will be happen? This both amino acid will make a bond formation. Okay, bond formation. The both methionine and tyrosine, it will make a bond with each other. I had told you that which type of bond they will make from the two amino acid. When the water molecule will be eliminated, so they will just join with each other. That one is called as peptide linkage. Okay, through the peptide linkage, both amino acid will be joined with each other to make a peptide, to make a new peptide one. Okay, now what will be happen? So now when they will make a new bond, so that will be our peptide chain will be formed, and now this tRNA, it will be shift to the E side through which it just exit. Okay, this tRNA, it will be out from this particularly ribosomal unit, and this process will be continue. It means now this particularly when they both will be bind with each other, this particular will shift to the P side. New tRNA will come here with a different amino acid. Now both these three amino acid will make a bond with each other. They will make a peptide formation. They will shift the P side, then through the E side, it will be just exit. Now again the new tRNA will come with a new amino acid. Like this process you can see. One is coming, one is going. Like in this world is happening. One is coming, one is going. Okay, everywhere. Okay, like this. Now what will be happen? The termination is when this particularly thing. Now when this tRNA is just coming, new tRNA is coming, and the process is continuing. But when this tRNA will just read this particularly our stop codon, as I told you, stop codon is UGA, UAG, UAA, and UGA. When this tRNA will read our this particularly codons, what will be happen? It will understand that now we have to stop bringing amino acid. What will be happen? This particularly termination will be formed. It means new peptide chain is already made, and now this tRNA will be just exit through this site, and this peptide, it will be just eliminate, okay, from that ribosome. Now we will see through the video. Now just see what is this. Wait for some time. It's taking a time. Yeah, this one is our messenger RNA. Why messenger RNA? Because you know this one is a single helix, right? This one is single helix. It means this one is our messenger RNA. It's having a this particularly cap. That cap is known as a methylated cap is there. Now they're having a here a chain molecule will be found after that. This messenger RNA having a codon. Now we will see the video. Now just see this one is our RNA having a direction 5' to 3'. Now in the starting they're having a poly A tail. Okay, this one is called as A tail. After this one, as you can see the difference, there is AA, but now there is something changed, right? What is this? There should be our codons, genetic codons. Now just see, this one is our messenger RNA codons. Different colors represent that there is different codon or there is a different amino acid. This one is our methylated cap. Okay. What is this? This one is our smaller subunit of ribosome. Now it will just read our initiation codon. Where it will read the initiation codon, on that side, this smaller subunit, it will be attached. Okay. Now it has just attached. Now this one is our tRNA. This one is our tRNA. So now what is this one? Red one. Yeah, this one red one. Red one indicates this tRNA is bringing any amino acid, and now this one is anticodon. Now just see, these anticodons are matching. As you can observe, these anticodons are matching. A opposite U, U opposite A, cytosine, guanine. Now our synthesis will be now start. Methionine. This it has bring a one methionine amino acid. Now it will be, yeah, just see, this one is a larger subunit has covered this particularly thing. As I told you, they're having a three site, A site, P site, and E site. So now it is in a P site now. Now new tRNA is coming with a new amino acid. As you can see, this orange is the new amino acid. The both amino acid will make a bond with each other through the peptide bond. And this tRNA, it will be exit. You see, this tRNA is exit. And now again, this one is acceptor region, peptide region, and exit site, like this. Now you can see all these amino acid peptide bond is making, and the tRNA is exit to the E site. A, P, E. And this process continues. That's why this one is called as elongation procedure. And interesting. So only you have to learn a small basic thing. That's just see, you will make a one movie, and your particularly biochemistry, this particular unit will create a new interest. Now when it will see this particular stop codon, it has seen this particular stop codon, what will be happen? Release factor will come there. Through the release factor, what will be happen? This tRNA, it will be just eliminate, and we will get our this particularly peptide. Protein. Okay. So like this, our this particularly translation is also finished. So only we have to focus on smaller things. So what will be happen this particular in the translation? Okay, transcription and DNA replication. So in the replication, we have seen in the replication 4 enzyme we have to learn. Already I have told you. In the transcription, you have to learn 2 enzyme. It's 2 things. That one, first one is promoter gene. The next one is RNA polymerase enzyme. The both things are sufficient. In the translation, you have to learn a small role. You have to learn the role of tRNA, role of A site, P site, and E site, role of codon, role of anticodon. And if you have learned all those things, then everything will be so easy for you. Okay, so if you have any queries, so you can ask me. The other thing is, I'm going to upload on your Moodle this video, and this lecture will be also uploaded, as well as the another thing is another offline video I will conduct today regarding your one chapter in this in the organic chemistry. Okay, that offline video I will be uploaded on your particularly Moodle. From there you can download it today. Okay, the rest thing you have to complete your practical file. Okay, and after completing the practical file, one day I will declare that now you have to show me the practical file only. Okay, then we will interact and we will see although, and then I will correct the those who have completed the practical file, then only those persons will get a marks, otherwise no. So please complete your practical file, as well as you have to just read, you have to prepare all your organic chemistry and this particularly chapter in your biochemistry. So hoping for the best and have a good day.